genetic information as shown in the leftmost cell. That is the flow of genetic information in cells. Okay, there is something called central dogma that says that the flow of genetic information goes from DNA, which is then transcribed, transcription, to make RNA, and then that is translated to make protein. Okay, and the central dogma states that the flow of genetic information is unidirectional. That is, we go from DNA to RNA, specifically mRNA, to protein. We call transcription by that name because we're going from one type of nucleic acid to another. That is, we're going from one nucleotide language, deoxyribonucleotide language, to another ribonucleotide language. It's very much as though we had taken our notes longhand in class, and then we went and transcribed them into computer font later in the evening. We're still in the same language, English, just different forms. Transcription. Translation, we'll find out, takes the information that is in ribonucleotide language, the mRNA, and translates it into protein. It's translation because we're going from ribonucleotide language in the mRNA to amino acid speak in the protein. So it's as though if we took our notes that we had in computer font and we ran them through a language translator and translated them from, say, English to a different language. All right, so looking at the structure of DNA, DNA is the polymer of nucleotides, A for adenine, T for thymine, G for guanine, and C for cytosine. DNA is referred to as a double helix, okay, because it consists of two helical strands that are tied together by hydrogen bonding. More about that later. Our double helix, so these two strands are going to form spirals, hence double helix, very much like a spiral staircase, okay, where the rail will be the sugar phosphate backbone, that is the phosphodiester bond portion, okay, and then the rungs of our staircase will be the nitrogenous bases, the adenine, thymine, guanine, and cytosine. Okay, and then of course we would have the assorted nucleoproteins, such as histones that are associated with it. Okay, as we said, the backbone is composed by sugar phosphate, and that's formed through phosphodiester bonds that are formed when we polymerize the original deoxyribonucleotide phosphates to make our chain. Okay, and then we'll have our second strand. The two are held together by hydrogen bonds. There is something called base pairing rules that say that if we have on one strand a thymine, then the other strand to match, there must be an adenine. So adenine pairs with thymine, guanine pairs with cytosine. This is the ATGC base pair construct. Okay, this is what's meant by the strands being complementary. That is, if we see an A on one strand, we have to see a T on the other. Otherwise, there's a mismatch, which could have dire consequences for the cell. Fortunately, we have mechanisms that can repair that. Anti-parallel. Okay, if you look on the leftmost strand, you'll note that the top portion of it starts out with the phosphate at the 5' end, and then when we go to the bottom, we have a hydroxyl, the 3' hydroxyl on the right, the deoxyribose sugar. That's the 3' end. The other strand on the right side is flipped. Okay, so now the 3' end is uppermost with the 5' end at the bottom. So, nucleotides have 3 ends. If we look at the top portion, we have a phosphate, 5', and then we have a hydroxyl at the 3'. Okay, note that the designation prime refers to the carbons in the 5-carbon sugar. Okay, so 1' would be the first one. Going clockwise, we would have 2', then 3', 4', and then 5'. So the 5' has the phosphate, the 3' has the hydroxyl. Okay, and at the hydroxyl, the 3' hydroxyl, is where we would add a new nucleotide if we wish to grow the chain longer. Chain terminators. Chain terminators are nucleotide analogs that have the same structure as a nucleotide, however, they lack that free 3' hydroxyl. So if we were to add a chain terminator into a growing DNA molecule, then because there's no 3' hydroxyl, we can't add any more. So we would wind up with shorter DNA than is optimal. That is, whatever organism or, for instance, virus incorporated the chain terminator, it would have too short genome, too short genetic information. It would lose genetic information, and so it would be defective. Okay, we use chain terminators, for instance, for anti-HIV medications. In fact, the first anti-HIV medication, azidothymidine, AZT, is a chain terminator. We also use them for treating certain cancers. Okay, and to recap, the structure of DNA is like that of a twisted ladder or spiral staircase, where we have the two helices, that is the sugar phosphate backbone, with the rungs, the cross pieces, being the adenine and thymine bonding together through hydrogen bonds, or the guanine and cytosine. Remember, we have the base pairing rules. A binds with T and G bonds with C. Also, remember that in order for us to replicate, to copy the DNA, we do need to get to the information that is contained in the nitrogenous bases, the adenine and the thymidine, or the thymine, and the guanine and the cytosine. Okay, and the way that we do that is to split the double helix apart. Remember that each nucleotide, deoxyribonucleotide, consists of 3 parts. We have the 5-carbon sugar, in this case deoxyribose. We have the 5' phosphate, and then we have the base, that is the nitrogenous base, either adenine or thymine or cytosine or guanine. Remember that the base contains the coding information, whereas the sugar and the phosphate are the scaffolding. That is, they are what are used in making the phosphodiester bonds that link the individual bases together. Also remember that we will be joining the 5' phosphate of an incoming base to the 3' hydroxyl of the existing base to form the phosphodiester bond. So again, DNA chains grow from the 5' to 3' direction. One cannot add bases at the 5' end of a DNA molecule. This figure shows a useful way of thinking about DNA bases, that is the deoxyribonucleotide triphosphates, as paper clips, where the yellow end is the 5' phosphate, the purple end is the 3' hydroxyl, and you'll note in image 2 that, or in panel 2, that we are always joining the 5' phosphate on the incoming base to the 3' hydroxyl on the existing base. And we just keep doing that until we make a chain of the desired length. It's very important that DNA replication be high fidelity. That means that there are very few errors and that errors are corrected. Errors, of course, are what we also call mutations. Potentially any change, particularly in certain critical genes that are key to cell function, have deleterious effects. That is, they're harmful. So we want to make sure then that we have high fidelity replication. And the key to that is that the DNA strands are complementary. That is, if we have an A on one strand, there has to be a T on the other strand. Okay. Also, replication is semi-conservative. We're going to make 2 double helices. Each one will have one strand from the original molecule and then one new strand. This is what we mean by semi-conservative.
All right, so we said that in order to do DNA replication, that we have to separate the strands of the DNA. And we do this at a special place on the chromosome called the origin of replication, ORI. Okay, and we see ORI depicted at the top right corner of our figure. So now if we go down, so we're blowing up the figure, we're expanding it so that we can see better what's going on, then what we've got happen is that at the right-hand side is where we started our ORI. This will be important later. That's where we opened the DNA and then we allowed the replication machinery to get at the DNA. Now, it turns out that, remember, DNA strands are built from 5' to 3'. Also, having single-stranded DNA exposed for any length of time is not good because it's unstable. It's prone to environmental attacks. So when the enzyme helicase, which is that blue triangle to the left, opens up the DNA at the ORI to generate the 2 single strands of our parent DNA. This is an area that's referred to as a replication fork. We need to cover the DNA as soon as possible. We need to make the second strand. For the leading strand, that's not a huge problem because we're going to make the copy, the complementary strand, continuously because we'll be able to go from 5' to 3' and we'll be going in the direction of the replication fork, which is to the left. That's all good. The enzyme that does the copying is DNA polymerase, proteins that protect the single-stranded DNA that's by the replication fork are called single-stranded binding proteins. Another wrinkle in DNA replication is that when we pull the 2 strands apart, that's going to cause the molecule to kink, to knot up. And so in order to relieve that tension, we have the enzyme topoisomerase or gyrase, which is the golden rectangle to the far left. All right, so we're all pretty good with the leading strand. It's the lagging strand where we're going to have issues. We're not going to make that one continuously, because if we did, we'd have to leave our single-stranded parent strand exposed for too long, and the cell doesn't have enough single-stranded binding protein to cover the whole thing. So what we're going to do is synthesize the lagging strand as a series of short pieces of DNA called Okazaki fragments. Now, it also turns out that we need to make a short RNA primer so that the DNA polymerase that's going to make the Okazaki fragment has something to latch onto. The enzyme that will make our little short RNA primer, RNA primase, or just primase, and then our DNA polymerase. And we said we weren't going to worry about whether it's DNA polymerase 1 or DNA polymerase 3. We're just going to say DNA polymerase. Our DNA polymerase will bind onto the RNA primer that was made by primase, and then it will extend that primer by adding deoxyribonucleotides to it. So we've made an Okazaki fragment. Now, let's look to the right, and we said that the replication fork was moving in the left, or to the left, and so we would have opened the DNA at the ORI, and so we would have made our first Okazaki fragment, okay, and we would have had DNA polymerase chew away the little RNA primer, and we can see that happening between Okazaki fragments 2 and 3. Now, we still have a gap in the sugar phosphate backbone, so another enzyme, ligase, which is the purple square, diamond-shaped, will come in to seal that.
This slide shows why, in better detail, we have to replicate the two strands of DNA differently. Okay, if we look up at the top, what we see is the beginning of the replication fork. Okay, that's where the DNA is opened at the origin of replication, ORI. Okay, and then we note that the leading strand, which can be replicated in the 5' to 3' projection, is replicated continuously. Okay, however, the second strand, the lagging strand, is replicated discontinuously. That is, we have the first Okazaki fragment formed where the ORI is first opened, okay, and the replication fork proceeding. And then as the strands are opened, we have the second Okazaki fragment being formed, and then that will be joined with the first using DNA polymerase to chew back the primer and replace those RNA bases with DNA bases, and then the ligase will come in and seal up the gap in the sugar phosphate backbone. This slide depicts some additional characteristics of DNA replication. 1. DNA replication is bidirectional. That is, we opened up the DNA at the ORI, the origin of replication, and then replication proceeds in both directions from the replication bubble. So we have two replication forks. The idea for this is that this would allow the cell to replicate the DNA much more quickly than if we started at the Ori and then just proceeded in one direction or the other. It's very much like the Transcontinental Railroad. Instead of building the railroad from the East Coast to the West Coast, what was done was to start at both the East Coast and the West Coast, and then complete the railroad by meeting in the middle. That allowed it to be built much more quickly to span the continent. Okay, another thing is that DNA is methylated. That is, methyl residues are added to either adenine or cytosines that occur in specific patterns on the DNA. Methylation plays a role in a variety of processes. In the prokaryotic world, cells use methylation to mark their DNA. That is, to know that their DNA is theirs and not that of an incoming virus. For instance, if a virus that's made through a non-methylating variety of bacteria, that is, one that does not mark its DNA with methylation, tries to infect a cell where methylation is going on and cells mark their own DNA, then because the viral DNA lacks the methylation, it does not have essentially the secret handshake, the code phrase to get into the clubhouse, and is destroyed. That is, this is a way that bacteria can keep themselves from being infected. Another thing that we do with methylation in eukaryotes, as opposed to prokaryotes, is we can use methylation to control gene expression. That is, whether a gene is on or off. Remember, by on, we mean that the gene is in such a way, such a configuration, that the transcription machinery can get in it and make RNA copies, and then those RNA copies can be translated into protein. Okay, so this is a way of turning genes on and off. Genes that are on are lightly methylated or non-methylated. Genes that are off, inactivated, are heavily methylated. Why do we care about this? Well, for instance, in the case of sickle cell disease, okay, people would have 2 copies of the sickle cell hemoglobin, which then renders them susceptible to crippling episodes where their cells sickle, because all they can produce is the sickle cell hemoglobin. That is, they're homozygous for the mutated hemoglobin gene. It turns out that we have 2 hemoglobin genes. We have adult hemoglobin genes, and we have fetal hemoglobin genes. When we're born, the fetal hemoglobin genes, which were active when we were in utero, are inactivated by methylation. They're still functional, but they have been turned off. If we treat people who have sickle cell disease, that is, who are homozygous for the sickle cell hemoglobin, what we can do is demethylate the fetal hemoglobin, which is functional, and this allows people who have sickle cell disease to produce a hemoglobin, fetal hemoglobin, that is much more functional than the sickle cell hemoglobin. This thereby mitigates the disease. That is, it makes the people better because they can make a more functional hemoglobin, and so their blood cells, their red blood cells, don't form the sickle cell shape that is the disease form.